Gracias. Buenas tardes a todos.
Very good afternoon to all of you, and to share our results before all of you. The presentation will be in English. I apologize for that. But it is perfectly possible to ask in Spanish if you need to do so.
Thank you for the invitation. Actually, I was a little bit surprised, but I'm very happy to show our results. And this is a picture of Munich, where our research center is located. Can we have, oh, I have this. I can do this. How does it work? Can somebody help me? Aha. Gracias. Okay, so Wilson disease actually, as you know, was first described by Dr. Wilson. That's where the name comes from. And here you see one of the very first patients he described that 1 year later, after this first picture, he was unable to write anymore and then couldn't walk. And the interesting thing in this original manuscript was that already there, Dr. Wilson cited a colleague from 1880 something who has described a similar case in a young boy. And already at that time, the clinicians thought there must be a toxin. This young boy must have suffered from an intoxication. And as you all know, today we know the toxin we talk about is copper. And how do you do the pointer here? The red button? Okay, gracias. So typically when we eat, the copper comes in from the stomach and it goes via this vein here, the portal vein, to the liver. And the liver then decides what to do, okay? So if we need copper, it brings it to the blood. If we have too much copper, it will excrete it into the bile and into the feces. And in Wilson disease, this central function is not working anymore. And that is the reason why the liver is of such fundamental importance in this disease, because the copper is accumulating there because it is not excreted. Now, we came across this disease because I'm the researcher, the lab rat, not the clinician here. And we came across this disease from this report from Jonathan Gitlin, in which he said, okay, here you see these liver cells. And again, copper comes in here in the liver, and then it is this green guy, this ATP7B that we heard a lot about before, who decides what to do with the copper, okay? So if we have too much, the copper will be excreted here into this bile caniculus and will go to the feces and bye-bye. Now in Wilson disease, this colleague here is not working anymore. And this causes the copper to accumulate in this cell. Now, what we saw also in this cartoon was this stuff here, mitochondrial damage. And that is why we got interested, Now I have to stress you a little bit with mitochondria. I apologize, but you will see it's worth it. So when you ask yourself, why does a cell, like the liver cell, needs copper? Why is it so important? The answer is easy. Because these little guys here, called mitochondria, they need copper for respiration. Every breath we take in oxygen, and from the oxygen we produce water, okay? And energy. And where is this crazy reaction happening? In the mitochondria. What a surprise. And how is this reaction done? By copper, okay? That's one part. There are other parts, but copper is highly important. Now, interestingly, in this disease, we have too much copper. So, why not look inside the cell where the copper is needed, whether there is an effect? And as Jonathan said, yes, there is one. And that's when we got interested. And you see this, so here's our lab rat, apart from me. And this rat has exactly the same mutation that Wilson patients have, okay? It has an ATP deficit. And when we look into the liver, into one of these cells, we do see all these very strange mitochondria. You will see immediately that they are not normal. You can realize, we call them in the lab the croissants. What is it, croissant in Spanish? Croissant, no? Okay, because normally they are nicely round. And here you see a liver picture of a Wilson disease patient. And you can nicely see, if you compare this structure here with this strange white area, that it looks exactly the same in the human patient and in our rats. And we then ask, okay, they look strange. And why do they look strange? What we realized was, when you take these mitochondria, you can isolate them, okay? And then you can study them. And that's how they look isolated. You see normal ones look round, and the older the Wilson animals get, the more croissant you have, okay? The interesting thing here is, when you measure the copper in these mitochondria, you can see that it goes up and up and up. And be aware, this is a logarithmic scale. So when the animals are about 3 months old, they have 10 times the copper compared to normal levels, okay? We also found, when the animals get ill, that this copper almost explodes to up to 100-fold, okay? And that is when the mitochondria look like that. And if you have something like that, the cell is dead, okay? Because it simply cannot produce any more energy, okay? Now, this is a normal liver mitochondria. And this is, we have around 2,000 in every liver cell, and they nicely produce energy, day in, day out, okay? Now, one thing that is repeatedly said in Wilson disease is, this disease is because of oxidative stress. Why is this? Because every chemist knows that copper can jump between two redox states. And when it does this in the presence of oxygen, very dangerous radical species may evolve. And that is called oxidative stress. Now, I was skeptic about this because it is also known that copper is not free. It is always bound. So we were very skeptic whether these structural changes, what you see here, are due to oxidative stress. So we did a simple experiment. We took mitochondria that look like this, and we treated them with copper. Okay, what a surprise. The result was, depending how we do the experiment, either with reducing conditions, so no oxidative stress, they look like this. Can you see the croissant? If we do the opposite experiment and we do copper with oxidation conditions, the mitochondria look like this. Completely different. And that was one of the major reasons why we said the oxidative stress comes late in this. It is not the reason for the damage, because in vivo, in our animals, in humans, the mitochondria look like this, and only if the animal gets really, really diseased, they look like that. So in the disease, we go from here to here to here. And this comes now with a very important consequence. The consequence is, if you want to treat this disease with antioxidants, it will not work, because it may block from here to here, but it does not block the way from here to here. What is the consequence? When we look at these mitochondria, and I told you they are responsible for producing energy. What would you expect? If they are damaged, you would expect they produce less energy. Huh? And that's exactly what happens. What you see here is ATP is the universal mark for energy. That's what they produce, which is equivalent to energy, cellular energy. And you can see when the animals are still healthy, they nicely produce energy. When the disease starts, it goes down, and then again another jump, and that is typically when these animals die. And you can nicely see this here at this first jump of mitochondrial damage, where you see a jump in energy production. That is when the animals get diseased. You measure this, we heard this before, in the blood by these liver enzymes. You can also measure the microRNA that Carmen just introduced to us. And then on the second jump, that's about 120 days of age, these animals are dead. All of them. Still today, if we don't treat these animals, we have not a single survivor. Okay? Now, the liver is not stupid. Huh? It realizes, hey, there is a problem. My mitochondria are not working anymore. I need to get rid of them or make new ones. How does the liver do this? The liver tries to bring these mitochondria into a sack, like a garbage bin, where it is degraded. Okay? Scientifically, we call this process autophagy, which basically means self-eating. Yeah? So we digest material, every cell digests material that is broken, that doesn't work anymore. And you can see this, I hope you can see it, that these are normal mitochondria. This is a control animal. These are Wilson animals. Here you can see the holes. And here you can see such a garbage bin with mitochondria inside to be degraded and to be renewed. This comes with a dramatic consequence. Because, let's say you have mitochondria that are not working anymore, and you try to remove them by bringing them into garbage bags. This could be very dangerous, because if you do this too much, you will kill your cell, because it doesn't have any energy anymore. Also, from this, you understand why this process, autophagy, is currently a very, very hot topic in research, because it also offers the idea to renew our cells, okay, to give them fresh energy. Can you block this process? The answer is yes, you can. If you eat, and we did this by high-calorie food. If you eat a lot of energy, which means a diet that looks like this, yeah? We all know it. Yeah? There's a lot of sugar, there's a lot of processed fat, there's much more energy in this food than we actually would need, okay? So if you give the cells a lot of energy, they don't need to make new mitochondria, because there's so much energy coming in that with even not so good mitochondria, they can happily survive. And biochemically, this means you block the process of mitochondrial renewal, okay? I told you before, the mitochondria are damaged, and this process may renew them. Now we stop this renewal. What will happen? The mitochondria should get worse. And this is exactly the case. And you see this here at these pictures where here it is again, the croissant. And if you see normally, these are our Wilson rats on normal diet. You see at 90 days they get ill and they die somewhere here. On this high-calorie diet, the disease starts already at 70 days, so much earlier, and it gets severe much faster. In fact, we had to stop the experiment here already at 80 days because these animals were so ill. Yeah? And the reason also was, when we looked then at the mitochondria and at their power, you can see they're somewhere here. So these are control mitochondria in producing energy. This is the fast food alone. This is the Wilson rat alone. And if you combine these two, it really breaks down. And this is actually one reason why we suggested to the clinicians to also consult the Wilson patients, not only for the copper content in the food, but also for other content in the nutrition, like enormous intake of calories, okay? Because we think from our data that this could be making things worse, okay? Now, I hope I could have convinced you a little bit that the mitochondria in the liver cells are highly, highly indicative of this disease. And I also showed you that simply treating oxidative stress doesn't work. So, what does work? And the idea is very simple. If we see that the mitochondria get worse and worse, and the copper increases and increases, we came to the conclusion the copper needs to get out. Okay? As simple as this. And the cool stuff here was we had a very little simple experiment. We could isolate the mitochondria. Let's see. So we isolate these mitochondria, heavily loaded with copper. We open our fridge, looking for compounds that bind copper. Where are you? Ah, here. Okay. Throw it on the mitochondria, steer it for half an hour, measure copper again. That's what we did. And we did this with compounds that you know. This one, penicillamine. I think you all know it. This one, trientine. Currently, the two compounds in clinics, right? We also tested many, many compounds, but I don't want to bore you with that. I show you one. We also use this one here, which is called methanobactin OB3B. What is this? It is a little compound that is done by this guy here. It's a bacterium. This bacterium lives on methane gas. Methane, that's the gas that comes out when we poop, okay? And cows and so. This guy lives from this gas. And it takes the methane and it does energy with it. And it does this by an enzyme, and this enzyme is a copper enzyme, okay? So in other words, this young fellow cannot live without copper. Where's the story? The story is, it also lives in surroundings where there's hardly any copper. So it has a problem. What does it do? It does this little molecule here, and it throws it out, and the guy looks for copper. Where is it? Ah, there it is. And it binds it, and the bacteria take it up again, digest it, and there they go, okay? Now the interesting thing here is, this compound has an affinity for copper that is by far higher to these two. Actually, it's by far higher than every enzyme in our body. That's number one. So it's very specific for copper. Number two, which actually is, in my opinion, even more important, it binds the copper and it stays mobile. That means it stays in solution, it doesn't react, the copper doesn't react, so it's chemically inert. It binds the copper, done. No side reaction, and no such thing as precipitation. Yeah? It stays in solution. And so we treated the mitochondria, isolated mitochondria with that stuff, and you can see if we do this for half an hour, no depletion with penicillamine, nothing with trientine. We also checked tetrathiomolybdate a little bit. And here's the methanobactin. So we said, okay, cool. And in the meantime, we have a new methanobactin, another form. There are several bacteria, and they produce different ones. We have this one, which is now with a company who tries to further develop this. It's called 101. And you can see we did the same experiment, and with 101, we can nicely deplete this. And when we do this, the mitochondria get better in energy production again, what you see here. Great. Then we said, okay, where does it go, the copper? It binds copper. Where does it go? And this we did together with Peter and the colleagues from Aarhus in our live rats. So here is a picture of the live rats. And what the colleagues in Aarhus did, they injected into the live animal copper. Okay? The copper goes to the liver, you see it here. And then they applied different chelators. First, buffer alone, so this is the control, nothing happens. This is penicillamine. You can nicely see, okay, it goes into the bladder. And here is the 2 methanobactins. And you can see within 20 minutes, the copper is going from the liver to the gut and getting excreted. And we then did, okay, if it goes to the gut to be excreted, is this happening in vivo? Does it really go out? So we put our animals into these metabolic cages. They sit here, and we measured the copper upon treatment in the feces and in the urine. And you can nicely see, here's 101, this is OB3B. Over days, they excrete the copper in the feces. And this comes with a dramatic reduction of the copper in the liver. This is the untreated animal, penicillamine, OB3B, 101. We calculated from here, this is 4 days treatment, 4 days. We calculated, okay, so if we come to half the value after 4 days, what happens after 8 days? We should be down at wild type level. And this indeed was the case. You can see it here. This is a wild type animal. This is the Wilson animal. This is Wilson gets ill, and this is treated for 8 days. No copper anymore. The copper is gone. We then thought, okay, if we can treat an animal for 8 days to reduce the copper down to wild type levels, we do what we do best, nothing. We go home, or we go to a fiesta, have a little party, and let the animal just live its life. And I tried to show this here by these graphs. So this is the lifetime of a normal rat, typically 2 years or something. This is our rats when they get ill, 90 days. This is when our rats die, around 120 days. Now, one treatment, what you typically do also in humans, is before you become ill, you start treating with penicillamine. Yeah? And then you stay on drug for the rest of your life. Same in the rats. So we came with the idea, hey, we treat for some days to deplete the copper, then we go home, do nothing, and when the copper goes up in the liver again, we treat again. Then we go home again, and so on and so on. And we did this experiment, and you see it here. So these green periods are the periods where we treat, and the red dots are when the liver values are going up. So you can see here they go up. Here we start treatment, poof, they go down. We go home, party, Oh, here they go up. We treat again. Poof, they go down, and so on, and so on, and so on. And by doing this, we could nicely keep our animals healthy and alive like a normal animal. Okay? You see this just here by the body weight. This black thing here is a control animal. These green ones are our Wilson rats. They nicely gain body weight. This one is an untreated that would die around here. And these animals happily lived a nice life. And you can nicely see here, we celebrated the first birthday of our long-term survivor because we were not allowed to bring in cupcakes in the animal house. It's not allowed. So we had this picture and played a little music, happy birthday song for them. And I think I rather stop here with the story because I wanted to show you much more, but in the case of time, I'd rather like to thank the people here. These are the colleagues in the US who do these peptides, these methanobactins, the people from Warsaw, from the clinics with whom we interact. We have the colleagues in South Korea who now try to bring these methanobactins into the clinics. We have the Aarhus people from Thomas and colleagues. We have the Munich people, that's us, and also work with Svetlana in the US, and Davis, and in Naples. Thank you.